So we're going to go ahead and finish up Chapter 16 notes. Remember, Chapter 16 is the innate immune system. We talked about the first line of defenses a couple times ago in lecture, and today we're going to finish up the second line of defenses. Remember, we've talked about inflammation and fever, phagocytes, and next up we have antimicrobial substances. So there's two antimicrobial substances we're going to be talking about. So the first one is the complement system. Then after we finish this, we'll be talking about the interferons. So let's go ahead and give a little background about the complement system. So the complement system is going to be this network, the system that is made out of proteins. So it's comprised of proteins that your body makes, specifically the liver. So made by the liver. And once these proteins are made by your liver, they're going to circulate throughout your blood and body tissues. We'll just put, so comprised of proteins that circulate. Let's put the body. So what happens is the liver makes these whole proteins. So like this here, these red ones, all these little blue ones here, these complement proteins. And whenever they're in their whole state, they're inactive, but whenever they get split, like here, they become activated. So I'm just going to put here that whole protein, the ones that are not cut or split, these are going to be inactive, whereas the split protein, the split complement proteins, mean they become activated. So let's put active. So let's go ahead and talk about this system and how it operates. So there's three ways that we can activate this complement system. So first up, we have what's called the classical pathway. So in the classical pathway, we're going to have antibodies right here. So here's our microbe, this brown thing. And in the classical pathway, we're going to have antibodies, so here, that are going to bind to an antigen on that pathogen. So antibodies will bind to an antigen. And when they do this, this is going to cause a complement protein to split. These then become fragments. You can see you get this cascade of reactions. And eventually, we're going to see that this protein called the C3, this complement protein 3, becomes split, which results in three different things. Okay, so we'll talk about that here in a second. So what are some other ways that we can get this activation? So a second way is we can have what's called the alternative pathway. So in the alternative pathway, they found the classical pathway first, and then the alternative way was the second way that they found. So once again, we have a microbe here, or a pathogen. And in the alternative pathway, I cannot spell. We're going to have these different protein factors here. So these are going to be like complement proteins. These are going to bind to glycolipids on the pathogen. So I'm just going to label this one here. So when these proteins bind to the glycolipids, that's going to cause the activation of the complement protein. So this one here is also C3, and this will get split into 2 fragments, resulting in the activation of the complement system. So we have those 2 ways. And let's look at the third way. So the last way is what's called the lectin binding pathway. So in this pathway, we're going to have these lectin proteins, these little blue kind of square looking proteins here. So this is a lectin protein. This is going to bind to mannose, so I'm just going to label this one here, on the surface of the pathogen. Right, so this was our, sorry, I should have labeled it microbe. So when lectin binds to mannose, that's going to cause this pathway activation, causing a response. So 3 different ways we can activate. Classical pathway, an antibody binds to an antigen of a pathogen. An alternative pathway, the protein factors bind to a glycolipid of a pathogen. And then for the lectin binding pathway, we're going to have lectin proteins that bind to a mannose on a pathogen. So what happens when these 3 different things happen? So eventually you can see all of them activate the C3 protein. When it gets split, we're going to have this 3 resulting events. So one thing that can happen is called opsonization. So opsonization is just a fancy term for coating the pathogens with these complement proteins. You can see here, here's the pathogen, these little red dots on there are complement protein. And these are kind of like handlebars on the pathogen. It's going to make the phagocyte grab on to the pathogen a lot easier when those complement proteins are present. So opsonization is just pretty much coating the pathogen with complement protein to increase or enhance phagocytosis. So I'm just going to put an arrow, increase phagocytosis. So it makes it easier for those phagocytes to grab onto and recognize those pathogens. So another thing that can result as an activation of this complement system is inflammation. So I'm just going to write that here at the top. So second result of activating the complement system is inflammation. So in inflammation, we're going to see that the complement proteins are going to cause these cells called mast cells to release histamine. So in inflammation, complement proteins cause mast cells to release histamine. Do you guys remember from the previous lecture that histamine, leads to inflammation? That's why if you ever have like an allergy or something where you get hives and you get real swollen and itchy, you can take an antihistamine like Benadryl to reduce your inflammation. All right, so we said we have 3 ways to activate the complement protein or the complement system. So what's the third result of activating the complement system? So the last thing we can do is down here at the bottom. We can get these complement proteins form this, it's called a membrane attack complex. And what happens is it forms kind of like this little tunnel in the plasma membrane of the bacterial cell or the pathogen cell, and it's going to cause that cell to burst open and lyse. So we'll just put here that you can get cytolysis occurs, and this is because the complement proteins are going to form a membrane attack complex, or a MAC. So that membrane attack complex, that's just, like I said, this little kind of tunnel-like thing that's formed in the membrane, causing that cell to burst open and lyse. So it looks pretty complicated, but for the complement system, just make sure you know the 3 different ways that the system can be activated. So where we had alternate pathway, classical pathway, lectin pathway. And, you know, as these activate the complement system, they split proteins to make them active. And you can kind of see the common denominator of all those proteins are the C3. These other little proteins have names as well, numbers, but you don't have to know those. But make sure you know that there's a common protein, the C3 protein, that when it becomes activated, you get 3 different results that occur to help, you know, clear that pathogen from the host. So we had opsonization, inflammation, and cytolysis that results from the activation of the complement system. So real quick, we're going to see that there's some bacteria that can actually kind of sneak around the complement system. So ways that bacteria can kind of sneak out of being harmed or being detected by the complement system are these. So let's put here's sneaky ways to evade or, you know, escape the complement system. So some bacteria will have a capsule. So if you have that nice, you know, coat, like a sugary coat around the bacterium, you're probably those different antigens on the surface of the microbe won't be detected as easily. So bacteria that have a capsule can, you know, kind of sneak around the complement system easier than those that do not. Another thing is some bacteria can have surface glycolipids. Some bacteria that have these surface glycolipids on their membrane, they're actually going to be able to prevent a MAC, that membrane attack complex that we saw over here, from forming. So if you have surface glycolipids, some of these can prevent that MAC from forming, so they can escape being burst open by that membrane attack complex. And then lastly, some bacteria can produce enzymes that digest complement proteins. So there's a certain protein called C5a that's going to be required to, activate this opsonization and inflammation. So if the bacteria can produce enzymes to break that down and make sure it doesn't allow, opsonization or phagocytosis and inflammation to occur, then they're going to be, able to escape the complement system a little easier. Not feel the full wrath of the complement system. All right, so we'll move on then to interferons in a second video.